So as before, the coat color of rabbits, multiple alleles can demonstrate a hierarchy of dominance. In rabbits, 4 alleles code for coat color. It's C, C to the CH, C to the H, and C. The hierarchy of dominance is as follows. The presence of multiple alleles increases the possible number of genotypes and phenotypes. Epistasis is an interaction where one allele hiding the effects of another allele. Seen in the coat color of Labrador retrievers, the dominant allele E determines whether the coat will have a dark pigment, while allele B determines how dark the coat will be. When a dog has a recessive EE or homozygous recessive alleles, the coat will be yellow, because the E allele masks the effect of the B allele. Sex determination. One pair of chromosomes, sex chromosomes, determine an individual's gender. A double X is female, and XY is male. The other 22 pairs of chromosomes are called autosomes. That's why we ask, is it sex-linked or is it autosomal dominant? Dosage compensation. The X chromosome carries a variety of genes that are necessary for the development of both females and males. The Y chromosome, however, mainly has genes that relate just to the development of male characteristics. In female, one X chromosome is inactivated in each cell. Called dosage compensation or X inactivation, which X stops working in each cell, is random. Here is an example of dosage compensation when we have a chromosome that's inactivated. Bar bodies. The inactivated X chromosome can be observed in cells. Darkly stained, inactivated X chromosomes are called bar bodies. Traits controlled by genes located on the X chromosome are known to be sex-linked. Because males have only one copy of the X chromosome, they are more affected by recessive X-linked traits. That's important to remember. Circle that part. For example, red-green color blindness is a recessive X-linked trait. The mothers are typically the carriers, and because of that, pass it on to their sons. So here the X is representing female, and the B is representing whether or not it's normal or has a color blind. As you can see, capital B stands for normal in our legend, and a lowercase b stands for color blindness. Okay, so if we look at the offspring where there are 4 available, we have a capital B and a capital B. So if we pretended these X's aren't even there, a capital B and a capital B would say that it's normal. A capital B by itself is normal. A capital B and a little b, it appears normal. However, for the genotype, we can see that this offspring will be a carrier. And then this offspring here is noted by just a small lowercase b. Because of that, this child will not be a carrier, nor will they be normal. They will actually have color blindness. Sex-linked traits continued. Hemophilia, recessive X-linked trait that causes delayed clotting of the blood. And in class we will talk more about Queen Victoria and her pedigree. In the meantime, be studying this chart because it will be on your test.
So the blood of a hemophiliac doesn't clot, and so they're very prone to bleeding to death. So this is the pedigree of one of the royal families of Europe. Queen Victoria here was the monarch of the United Kingdom from 1837 to 1901. And hemophilia acquired the name the royal disease because of the high number of descendants of Queen Victoria who were affected by it. So if we look at the key over here, we have unaffected male, We've got hemophiliac, which are all males because this is an X-linked recessive disease. And there was no females who had a father with the disease, so there was no chance of any females having the disease so far in the pedigree. So all the hemophiliacs are male. Then we've got the possibility of an unaffected female or a carrier. Okay, so if we look back in history before Queen Victoria, there are no known hemophiliacs in this royal family. So the assumption is that the hemophilia mutation occurred in the DNA of Queen Victoria's father's sperm. So she was the first hemophiliac in this royal family. So she was a carrier because, remember, for females, they would need 2 affected X's, since it's X-linked recessive disease. And so here are her children, and she kind of got unlucky here because she had 4 daughters, and 3 of those ended up being carriers. So we've got Victoria, Alice, and Beatrice, who are carriers, and then she had a son, and the son was a hemophiliac. So he also passed that trait. So all of the descendants here actually got the first line of descendants actually got the allele. Okay, so then if we follow it down, and Leopold died, so you'll see most of these hemophiliacs died pretty young. He actually was one of the longer living ones. And especially if you think, back, we're talking late 1800s, not the best medical care. So for someone with hemophilia, they're most likely not going to live very long. Okay, so if we look down to the next generation, so this would be essentially Queen Victoria's grandkids. So again, we've got, actually in this generation, all of the females are carriers, and we've got 4 affected males who you can see, again, all died young, and then we've got 4 unaffected males. It was very common in royal families for cousins to be married, and so that can actually sort of concentrate the allele. And so we've got, like this here, this is cousins getting married, and so you can see that they actually end up with both children having hemophilia. So now we're on to the great grandkids. This guy down here was murdered, and maybe he was more likely to die because he had hemophilia, so he bled more easily. And then all of the great grandkids here were males and ended up with hemophilia. And then it does actually say she had more children than what are shown here. So it's got 6 of the 34 shown. So there was more, but this is just to show you some of the...
So as we were talking the other day in class, if you can see down here at the bottom, now that I paused it, you can't see it, but right here, Rupert, it says, it should have, that's why we use numbers. That's why we say 1, 2, 3, 4, 5. So that way you can tell we've skipped something if it goes from 1 to 3 to 10. Okay, so that's why it's important to number them and not name them, so we know if there are people missing. Polygenic traits arise from the interaction of multiple pairs of genes, include such traits as skin color, height, and eye color. Environmental influences, sunlight, water, temperature. Without enough sunlight, most plants will not produce flowers, as you already know. Insufficient water causes plants to drop their leaves. And most organisms, including humans, experience phenotypic changes when they have extreme heat. On Google Classroom, I am going to have an assignment here very soon about this topic, twin studies. Twin studies help scientists separate genetic contributions from environmental contributions. Traits that appear frequently in identical twins are at least partially controlled by heredity. However, traits expressed differently in identical twins are strongly influenced by the environment.